Okay, so welcome to this next video in which we are discussing neurotransmitter release. So far what we've discussed is that down the axon will come these empty vesicles which we are going to fill up with neurotransmitter. Then we've discussed the process by which you can put neurotransmitter into these vesicles. And this is through a secondary active transport process. So you firstly use the vesicular ATPase, the VATPase, to pump protons into the synaptic, well, into the vesicle at the moment. It's not a synaptic vesicle yet. It's having protons pumped into the vesicle, and that's going to establish a concentration gradient where you have an extremely high concentration of protons in here. So I have to note the protons in blue. So you get an extremely high proton concentration in this vesicle. And then what's going to happen is that you can then use your neurotransmitter transporter to couple the movement of protons out of the vesicle to the movement of neurotransmitter into the vesicle, and that's how you can move the neurotransmitter into the vesicle, basically. And examples are this VGAT, the vesicular GABA transporter, which will move GABA in, coupled to the movement of protons out, and then the vesicular monoamine transporter, blocked by reserpine, which moves dopamine, serotonin, noradrenaline, the monoamines, basically, into synaptic vesicles. Finally, in the previous video, we discussed the reserve vesicle pool, where we have these synaptic vesicles stored away from the active zone of the axon terminal, and those are bound basically to the actin filaments of the actin cytoskeleton of the cell via these synapsin proteins here. Okay, now what we want to next discuss is how we can dock vesicles at the active zone membrane over here. Okay, and for that, basically, we need to discuss the snare proteins. So, do I have enough space to do this down here? I think we might have a go. We can have a go at least. Right, so, basically what's going to happen is we're going to produce these synaptic vesicles, which are these vesicles full up, filled to bursting with our neurotransmitter contents. And we now want to dock them at the membrane. So we want proteins in the membrane of the synaptic vesicle to interact with proteins on the plasma membrane and hold the synaptic vesicle at the plasma membrane, ready for if an action potential comes in, then they can fuse. Now, this is going to be a difficult question to tackle because something stops them from actually fusing. Now, usually what happens is when these proteins in a vesicle join with proteins in a plasma membrane, let's say, usually what would happen is they'd start fusing. These specific proteins that we're going to look at are snare proteins. So it is still a mystery what stops these snare proteins from fusing together initially, and why they only fuse when calcium comes in, as we're going to see, when an action potential arrives. Obviously, it would be a disaster if that didn't happen, i.e., if our neurons were just releasing these vesicles all the time. But the mechanisms underlying it are still controversial. We will look at theories, but it's still an area of active research. Okay, so firstly let's talk about the docking. These proteins in the vesicle membrane that are going to interact with proteins on the plasma membrane. Okay, right. So these proteins that are in both membranes are referred to as snare proteins. So snare, firstly, what does snare stand for? Well, snare stands for snap receptor. So snare proteins, when we talk about snare proteins, it means snap receptor. So the SNA comes from snap, and then the RE comes from receptors. So these are snap receptors, basically. Now, you can then ask, it's like a continuous cycle, basically, of pushing it back. I've pushed it back one level now. You can then ask, what does SNAP stand for? And we will see what SNAP stands for later. It stands for soluble NSF attachment protein, but at the moment that doesn't mean anything to you. We will see why these are called SNAP receptors later, and it was because they were first discovered to be binding to SNAP. Now, SNAP, again, SNAP's role in all of this is controversial. The theory I'm going to propose to you will have SNAP, we'll talk about SNAP much later, basically. Okay, right, so snare proteins then. So, the snare proteins can be divided into two main classes. The V-snares, okay, which are the snare proteins which are in the vesicle. So this just means vesicular snares, okay? So just like VATPA stood for vesicular ATPase, V-snare stands for vesicular snare. And then the T-snares, which stand for target snares. And in this case, the plasma membrane is viewed as the target membrane. So that's why the target snares are within the plasma membrane. So target snares. Now, in the case of the snare complexes formed in neurotransmission, what's going to happen, basically, is that one alpha helix is going to be provided by the V-snares, and 3 alpha helices are going to be provided by the T-snares. And if you don't understand what that means yet, don't worry. I will say it again in a moment, once I've actually drawn a picture. Right, so let's talk about what proteins in the vesicle are actually going to interact with which proteins on the plasma membrane. And I should just label this up. This is the plasma membrane, PN for short. Now, there is a protein in the membrane of the vesicle known as synaptobrevin 2, specifically synaptobrevin 2. Now, there are many different types of synaptobrevin. The one that's involved in neurotransmitter release is specifically synaptobrevin 2. However, you will often hear people just referring to this as synaptobrevin. It's the most important one, and certainly it's not essential knowledge that you know that there's the 2 afterwards. But specifically, this is synaptobrevin 2. Okay, so synaptobrevin 2 is the V-snare, basically. It's the snare protein that is in the membrane of the vesicle. Okay, now, we have 2 more snare proteins, which are in this case the T-snares now. Okay, so another snare protein is in the membrane of the, in the plasma membrane, and this is known as syntaxin 1. Again, most people will just refer to this as syntaxin, but there are many different types of syntaxin. When you study the fusion of, when you study different vesicle fusion events other than just the vesicle fusion events that are in that take place in neurotransmission, syntaxin shows up everywhere, and there's loads of different types. The syntaxin that's involved in neurotransmission is syntaxin 1. As we've seen in the anterograde and retrograde transport between the ER and Golgi, there are syntaxins involved there as well, and those are different syntaxins from this one. Okay, so syntaxin 1 is this snare that's in the plasma membrane and is involved in the formation of this complex between these proteins that are on the vesicle and the membrane. Okay, and finally, there is then a final one, which I'll draw like this, because it provides 2 alpha helices. So these lines here, they represent alpha helices, which are going to all interact together and form like a bundle. Okay, so this final one here, which I'll denote in blue, which has these 2 alpha helices, as you can see, this one is a protein known as SNAP25. Okay, so this is SNAP25. Now, what happens first then? Okay, so first, what's going to happen is that you're going to get syntaxin 1 and SNAP25, which are both T-snares, they're both target snares, meaning that they're both in this plasma membrane. They will associate first, and they will form complexes, basically, where the alpha helix of syntaxin 1 wraps around the alpha helices of SNAP25. Then what will come along is our synaptic vesicle, which I'm just going to make it more obvious that it's a synaptic vesicle by filling it with neurotransmitter. Our synaptic vesicle will come along to this plasma membrane, and then it will give up its synaptobrevin 2 here. So synaptobrevin 2 will now start also add in to this complex, basically. So you're going to firstly associate syntaxin 1 with SNAP25, and then synaptobrevin 2 is going to come and join the fun, and it's going to wrap around too. So they all have these alpha helices. SNAP25 has 2 alpha helices. So they'll wrap around each other and form this very, very tight bundle, which will bring the two membranes very, very close together. And that's a difficult thing to do because if you remember, the structure of these membranes is that they have phosphate heads sticking out, basically. So the phosphate heads have a negative charge. So this membrane is negatively charged, this membrane is negatively charged. You'll try to bring those two together. It's not going to work. But that's the feat that these snare complexes, and this is called a core snare complex. I should have that up here. So this is a core snare complex. These 4 alpha helices entangled with each other, basically, is a core snare complex. Specifically, at the moment, it's what's known as a trans core snare complex, because trans means on opposite sides. So you have the snare proteins, the V snares and the T snares, are on opposite membranes at the moment. As we see, once these actually fuse together, all these proteins are going to then be in the same side of the, well, they're going to be in the same membrane effectively. Okay, and then it will turn into a cis core snare complex, because the snare complexes don't suddenly just break down when the membranes fuse, they remain together. But at the moment, whilst you've still got the vesicle membrane separate from the plasma membrane, it's called a trans core snare complex. Okay, now it's a mystery basically as to why the formation of these really tight bundles, which are going to move these two membranes very close together, doesn't cause the fusion. But it doesn't, and I will describe to you a theory called the clamp theory, which explains why that, well, attempts to explain why that is. But as I say, this is a theory. It's not rock solid fact. Okay, this bit is very well accepted. This bit is solid. Well, obviously no science is ever solid, but this is more solid than the clamp theory, certainly. Okay, now these trans core snare complexes, you don't just form one, you form multiple. So there'll be another one down here. Okay, so you'll get lots of these complexes forming, which will all be pulling the membranes together, basically. So I'll draw another one down here, because that generally is how you'll see it drawn in pictures. It looks quite nice this way. So you have this synaptobrevin 2 here, forming this complex with the synap25 in blue here, which provides these 2 alpha helices, and then syntaxin 1, which provides only 1. And I have drawn them all in this parallel way, where all of the alpha helices are just sitting parallel. That is because it makes a nice picture. If I draw them all twisted up, it would look very, very complicated and very, very intimidating. You'd be trying to follow them, trying to understand what has he drawn. So I've drawn them parallel so that it's easy to understand, but in reality they're going to twist up together, and that's going to pull these two membranes together. Right, so this is how the vesicles dock. This is not how the vesicles fuse. And as I just alluded to, it's a mystery as to why these snare complexes don't cause the vesicles to fuse together. In other vesicle target membrane fusion processes, such as the anterograde and retrograde pathways, I described to a process where we formed complexes just like these, and those cause the fusion of the membranes. So what stops the fusion of the membranes in this case? Well, one theory is that you have a protein sort of sitting in the middle here, holding the two membranes apart, basically. And this is the clamp theory, basically, so I'll put that down somewhere. So the clamp theory, and I want to stress, this is not rock-solid science. This is a theory, this is a hypothesis, which has some weight behind it. Okay, so the clamp theory is that there is a protein sitting between these two membranes, which I'll draw as this green structure here. And this protein is basically holding the two membranes apart and stopping them from fusing, and stopping the core snare complexes from twisting enough that they bring them closer together. So basically, if you were to remove this protein, the name of which, I should give you the name of it, the hypothesized protein that does this is a protein known as complexin. Okay, so if the hypothesis is that if you removed complexin, what would happen is the core snare complexes would tighten up even more. Those would bring the membranes closer together, and then they would fuse. So the hypothesis is that you have this protein complexin that is sitting in between the two membranes and holding them apart, basically. And in accordance with this theory, we will see how when calcium goes up in the vicinity of this docked vesicle, it's going to cause the removal of the complexin protein, and then what's going to happen is these two membranes are going to fuse. That's the theory, anyway. Okay, right. So, just to summarize before we end the video, we have these core snare complexes, these trans-core snare complexes forming between the V-snare, synaptobrevin 2, in the synaptic vesicle membrane, and the T-snares, syntactin 1 and SNAP25, which are in the membrane of the plasma membrane. Okay, and those are what are holding these synaptic vesicles, which are in the readily releasable vesicle pool, at the plasma membrane, and they are waiting to fuse. They are not yet fusing, and the theory for why that is that there is this clamp protein, complexin, holding the two membranes apart and stopping them from fusing. And we'll continue the story in the next video, where we're going to look at how an action potential is going to bring about the fusion of these two membranes.